Hello everyone, my name is Chloe Hunt and today I will be giving my review on the ASM podcast Masterful Subversion. Michael, who currently serves as the chair of Council of Microbial Sciences, which is an advisory body to the American Society for Microbiology's Board of Directors, where they, along with the American Academy for Microbiology, are the board's, if you will, think tank for all things microbial. It's a big group with lots of expertise and insight where they are comprised of 35 branch counselors and 27 divisional counselors, representing all the scientific disciplines of the field in biology, immunology, and virology. They came together to figure out how Salmonella injects a protein into the cell to drive suppression of the immune response. Very important pathogen. It causes roughly around 150 million diarrhea illnesses a year, 600,000 cases of bacterial infections, and over 25 million cases of enteric fever every year, with the highest morbidity of any foodborne pathogen. Part of the reason why it's so successful is that it injects into cells effector proteins that modify the cell so that the cell now is a wonderful environment for itself. So this Salmonella has two type 3 secretion systems which deliver proteins. These are called your effector proteins. They're basically proteins that have an effect on the host cell. They manipulate the physiology and they allow the invasion of the bacteria. They establish a niche so that the bacteria may multiply. Salmonella enterica modifies the host defenses, so they're basically going to tell you a story of how a salmonella protein called SAR promotes an anti-inflammatory response. So what does that mean? Well, when you're infected with a bacterium or a virus, there are proteins in cells in our cells that sense the infection and they mount an antimicrobe defense, which involves production of cytokines, and these recruit immune cells into the area to get rid of them. But in the process, of course, it causes inflammation. You get swelling and redness and pain. But in the end, the idea is to cure the infection, and one major way that this happens is by pili cells binding to receptors on cell surfaces, and among the cytokines that are produced is one called IL-6 interleukin. This is what we call a pro-inflammatory cytokine. Interleukin 6 is made early on in virus and bacteria infection to help recruit all these mediators into the area and help clear the infection. IL-6 in itself can bind to receptors on host cells, and that binding is what induces the production of more cytokines. But then it also induces the production of regulators that turn off the pro-inflammatory cytokine response. That's because you don't want to have inflammation forever. It's not really good for you over the long term. The pathology of the SAR COVID-19 is due to treat patients with that to try and dampen down the inflammatory response. So what Salmonella SAR does is completely turns on the suppression part of that pathway so that all the inflammatory responses are suppressed, and that's good for the bacteria. Then, because there are no immune cells coming in to get rid of it, and it can multiply as it takes advantage of an existing pathway for suppressing inflammation and turns it on. The IL-6 receptor and that binding causes phosphorylation of the cytoplasmic portion of the receptor, which then recruits a cell protein called STAT. The 3-step is phosphorylated and it goes in the nucleus and turns on the transcription of genes that are involved in anti-inflammatory responses. So this SAR protein has a sequence. It has a 39 amino acid sequence that looks an awful like this cytoplasmic domain of the IL-6 receptor. So they find out eventually that this part of the SAR protein will eventually and actually bind to the STAT3. So the member statuary is normally binding the cytoplasmic domain of this IL-6 receptor, which is improving the implications of their vaccine that is in the works. Now, this bacterial protein known as RA is binding to it instead, that is now going to rewire. The response is to be overproducing these suppressors of inflammation. So just by looking at the sequence, they can identify that this is a 39 amino acid domain. They do some experiments to show that the protein will bind to the STAT protein 3. And that suggests this mechanism can functionally substitute and actually does a better job at turning on this pathway than the normal pathway does. It turns out that this SAR protein is phosphorylated at a specific tyrosine residue, and they show by knocking out that amino acid that the phosphorylation is important for the activity of SAR protein that is binding to STAT3. Binding to STAT3 would then lead to phosphorylation of STAT3, and we will then go from there into the nucleus and turn on all the anti-inflammatory pathways, so that phosphorylation is crucial for this activity. This protein binds with very high affinity to STAT3 and even more high affinity than this plasmic domain of this cell surface receptor. The IL-6 receptor that we've been talking about so much can also take a protein, and they do what's called the discovery screen. This is where they make the protein and they add it to cells, and they ask what other proteins are binding to it, and they identify them by mass spectrometry. And then among the proteins they find what are a couple of other protein kinases in the cell, and they are called the GSK3A and GSK3 beta. What's really interesting is that they eventually showed that the activity of one of these is absolutely needed for the activity of the SAR protein, and that is the ability of the SAR to bind to the cytoplasmic port, which then binds to the STAT3, and it is phosphorylated. And apparently the GSK is what is phosphorylating STAT3, because SAR protein doesn't have any kinase activity on its own. So here we have this model in which the bacteria injects into the cell, and the SAR protein binds to the cellular GSK3. It then phosphorylates the SAR protein, and then that results in the binding of the STAT3 protein, which ends up being phosphorylated, probably by the GSK, and it goes into the nucleus and turns on all the anti-inflammatory genes. So normally, again, to summarize, this IL-6 would bind to the cell surface receptor of the cytoplasmic domain and would be phosphorylated, that would bind to STAT3. STAT3 would be phosphorylated and would go in the nucleus, but now these bacterial proteins are taking the place of the cytoplasmic domain itself, which binds to the GSK3, and that binds with an even higher affinity to STAT3 than does the cytoplasmic domain of the IL-6 receptor. Michael and his team came together to convene a global summit on this COVID-19 topic. Specifically, where Marshall and its collective expertise and the ones of the broader AMS membership and staff would help contribute to the scientific efforts aimed at addressing this global threat, rather than reviewing results that were being deposited daily. This output of research results from the colleagues in China and from all across the globe. They thought that what this global summit might be able to help was to have a thoughtful distillation of the perspectives of these experts, so that they may come up with a series of coordinated global research priorities. They also provided the panelists with the WHO roadmap on COVID-19, which curiously was developed in 2016, but rather than naming it COVID-19, it was Virus X. And as you can imagine, they began to work through the planning documents and began to think about things they had lots of discussions on. One of the interesting things was that they really wanted to understand how they could help marshal the efforts of the entire scientific community and how to coordinate, maintain, and manage those activities.